So the meeting is now being recorded. Yes, welcome all of you to this 262nd edition of Thursday Musings. Today we'll be going a bit into the basics, functional architecture of the brain by Dr. Ritvik. The meeting is being recorded to be put on YouTube channel later on. Questions are invited in the chat box, so you can type your questions, which will be taken up by the presenter. Everyone will be on the mute mode. Next slide, please. So the chairman of this program is Professor Dr. Tufan Fatih sir. Over to you, sir. Next slide.
Thank you, Dr. Alim. Welcome everyone to this edition of the Thursday Musings. Next slide, please. I welcome to this and appreciate the contribution of our vibrant moderators, Dr. Alim Siddiqui, Dr. Amrita Patudusi. I think he has joined. Dr. Shobit Garg and Dr. Bishal Chabra. Welcome everybody. Now the introductory slides, please. Next. Shreya,
please change the slides.
Chairman's introductions.
So I think
Sir, I've changed the slides. Is it not?
it's not visible. We can see only Dr. Tufan's slide.
यहाँ से रुक गया मेरे से.
Sir,
Only Vishal bhai, can you download and share, because I think the connection is poor.
Let me
My
do it.
connection is poor and the
No,
voice is not.
I will do it. Just the musings. Give me a second.
oh, sir.
Can you unshare and share it? Can you put it on the? It's not on the chat also.
Oh, it is
Chat
not in the
नहीं है.
chart. I was looking for that.
आप दोबारा try करना एक बार, Shreya. Maybe you'll be able to do it.
Sure, actually. 1 minute.
Maybe.
Now, sir, is it visible? Now, sir, is it visible?
No.
Let me do one thing. I think I've got older slides. वो try कर लेते हैं एक बार.
अच्छा. श्रेया किसी के पास भी भेज दो अपना. विशाल.
Yes, sir. एक minute. Sir,
I have let me try and share.
Yes, sir, it is sharing. Yes, it is sharing. Yes, it is sharing.
Others, let me share what the slide I have, but it is not the same format. Can you see this slide, sir?
Yeah. And this will do. One by one you share, I shall tell.
Yes, sir. I'm going to. So this is Professor. So you please go ahead, sir.
I have the pleasure to introduce Professor Sri Gopal Goyal, who is at Department of Psychiatry and Addiction Demands, Prince Vijay Singh Memorial Medical College, SP Medical College, Bikaner. He is the professor and head of the department. His areas of interest involve adult psychiatry, addiction psychiatry, child psychiatry, neuromodulation, and polysomnography. Major achievements and contributions in the field of mental health, MD/MS thesis award, award for the best paper poster, Dr. Gellert Award, Rajasthan Cycle 2014, best poster paper award, Asif Nath Jod, Dr. Bakshi Award,
Please,
Asif Nath Jod, Dr.
let me
Solanki
begin,
Award,
फिर मैं call करता
Raj
हूँ.
Cycle 2018, Jaipur Award, Raj Cycle 2021. Contributed in IPS guidelines for substance use disorder. Published research paper in national and international journals. Contributed in training of field worker in recent national survey and of substance use in India and coordinated by NDDTs. Welcome, Dr. Sri Gopal. Next slide, please. विषय दूसरा है Dr. Parag Shah, Professor and HOD Psychiatry, SMIMER Medical College, Surat. Teaching, training, research experience of 22 years. PG teacher and examiner in MD, DNB Psychiatry, MCI, NMC, DNB Assessor, Direct EC member of IPS, EC Coordinator IPS, Accreditation Clinical Psychiatry Guidelines, Subcommittee on Child Psychiatry, Interpersonal Transcultural Psychiatry, CVT Specialist Section. Vice President cum President-elect IPS Vision, ex-President and ex-General Secretary, ex-Editor IPS Gujarat Statements, a National Advisory Board Associate Editor of Psychiatry Journals, Chair, Advisor and Panel of multiple government departments and non-government organizations for mental health issues including addiction, suicide, etc. With this, I welcome both the chairpersons and request one of them to introduce Dr. Parag Shah, sorry, Dr. or Speaker, Dr. Ritik Singh. आगे
I
चलो.
would request Dr. Sri Gopal to please introduce the speaker for today.
And with this, I hand over the meeting to the chairpersons. Kindly take over. And
Good evening, sir, and good evening to all the participants. Our speaker for this webinar is Dr. Ritvik Chatterjee. He is MD and postdoctoral fellowship in child and adolescent psychiatry from TNMC and BYL.
So you're muted, please.
Rishi, you got muted. Unmute करिए.
Am I audible?
Yes.
Yes,
Yes. Yes, sir. He is the winner of the J.C. Marpatia Award for Best Clinical Research in Child and Adolescent Psychiatry in India in Biennial Conference of Indian Association of Child and Adolescent Mental Health in 2019. Founder of Arambam Mental Health Clinic in the Pune. He is a consultant child and adolescent psychiatry in Minded Tilak Road, Pune. And he is a consultant child and adolescent psychiatry in Mind Smith, Gurgaon. I over to the our next chairperson regarding this topic. That is today's topic is the functional architecture of the brain. That is referred to the functional connection among the brain to various cognitive functions, emotionals, and the memory related functions. And I hand over to the my other chairperson, Dr. Parag Shah, for detailed introduction of the topic.
Thank you. Thank you, sir. Today's topic focuses on the functional architecture of the brain, something so basic and integral part of every postgraduate training, now essentially being recognized by all of us. And I'm sure it's also something very important for all of us senior psychiatrists to revisit our basics in psychiatry especially when we are moving more and more towards the neuropsychiatry aspect of our field. Friends, functional architecture of the brain refers to understanding the concept of the brain aspect of the mind, typically which we used to understand for many decades as the core functions of the mind are now being largely understood and considered important and integral for understanding psychiatric disorders from various network science perspectives. So today's topic and discussion focuses on models like how we understand modular functions of the brain, which means functional centers in the brain and how they are integrated with each other. A very interesting and important network science, which means various parts of the brain have nodes or areas performing specific functions, which are connected through edges, which are connections. And these connections build up, along with the nodes, a display of network. And there are various such networks in the brain which take care of various functions, typically till now, which we were understanding as functions of mind. And a very interesting aspect is that these networks are very dynamic and adaptive throughout the life, and they keep growing and changing. And that's how we understand the pathology and the recovery in psychiatric conditions. We are all very excited to listen to a very learned speaker tonight. And I'm sure we are going to learn a lot about various networks. For example, something which we have always heard like default mode network, salience network, and so many similar things which are going to be useful for us to rebuild our basics and concepts of neuroanatomy and neurophysiology. Thank you so much, and I welcome you once again to this forum and this session for today. I hand over the session to the speaker for tonight.
Thank you, Dr. Pawar. Uh, so, first of all, before sharing my screen, I want to talk to you guys about why this topic is important. Uh, Medicine currently is undergoing a very rapid change in our understanding about diseases. So, one of the first fields that got affected was cancer. So, in onco, if you by the time I have finished my MBBS, at that time we used to talk about things like squamous cell carcinomas and subtypes or adenocarcinomas. But now the current understanding is whenever anyone has a cancer, we do tissue sampling, we send for molecular targets for them, and with that we are able to make a specific treatment guideline for that specific person. So every single cancer now is gone into what we call as the precision medicine zone or personalized medicine zone. The advantage of this is so many new drugs are coming in onco. Currently, right now, onco is one of the largest areas of research because we understand things from basic to advanced. So this is basically what I would try to share with you, my understanding of what I would say how the brain works. And with that, I'll just share my screen. Sorry. Yeah. Are the slides visible?
Uh,
Yes,
yeah,
yes.
yeah, we
Yeah,
can
great,
see it now.
great. So I'll start. Yeah. So nowadays, our previous understanding of neuroanatomy, neurophysiology has basically been flipped over what we used to think. And we are looking at a hierarchical system in the brain. And you can think of it as four different layers that uh, interact together with each other. And this is what we'll be focusing on more. So the first layer is actually the molecular foundation. This is the multiomics network which form the brain's dynamic source code. The next one is synaptic architecture. So neural circuit processes and how they store information through plasticity. The third layer, Dr. Parag Shah already talked about this, is the bioelectrical grid. So these are oscillatory patterns which coordinate global brain activity, and these are what we measure as different networks. Finally, we have what we call as the brain-body interface, which mainly consists of brain controlling the functions of the body via autonomic, immune, and endocrine system. So we'll start with each layer and build up on top of it. So starting with the first layer, which is the molecular foundation or the multiomics. So we all understand that genomes leads to epigenomes, which leads to transcriptomes, after which there are proteins which are formed and they actually do end up doing the most of the cellular work. And then there is metabolome. So in each and every single cell, we are looking at least these 5 areas when we are talking. So multiomics integration is something which has changed the way that we look at how diseases occur and um, what kind of connections we are looking at. And one of the most interesting part about multiomics has been the single cell revolution. So previously, till this point, we used to think of things as basically the brain as bulk tissues, and we used to traditionally analyze at least signals from 1,000,000,000 to maybe even more cells, and we would average all of those, and this would leave out a lot of critical details. So right now we have single cell multiomics techniques like SNM PAT sequencing technology. Here, what happens is you're simultaneously profiling the DNA, uh, the RNA, as well as proteins, all the different omics that we talk about in a single nucleus and a single cell. So this is letting us understand what exactly a cell does and what kind of transmission it has with other cells. And it's a very specific kind of a function that we are looking at. Once we were able to look into the multiomics side of it, a lot of cellular diversity has been revealed. And we can look at different subtypes based on unique molecular signatures. And or we can also connect specific molecular patterns to distinct functional roles within the neural circuits. So first is the social network of cells or the intercellular community. So we have stopped previously, we used to think about how axons lead to dendrons and the signal is transmitted. And it used to be a unidirectional flow. This was what I was taught in my MBBS. But now we are currently looking at what we call as the tripartite synapse. In tripartite synapse, you are looking at the communication between one astrocyte, another astrocyte, as well as one neuron, another neuron, as well as the third will be a glial cell. And the glial cell basically coats the synapse. And we are looking at the glial cells taking an active part in this synaptic transmission. So these glial cells sense neuronal activity. They release neuroactive gliotransmitters. They can modulate synaptic strength and neuronal excitability, and also function as a computational element in neural coding. So what are the different styles of communication between cells? So the first one we are very clear about, ligand receptor signaling. So specific ligands are sent and they attach to specific receptors on the other cells and cause some cellular mechanism to take place. Except for that, there is also extracellular vesicles. So in extracellular vesicles, what the cells are doing is basically the kind of creating something like a small email and then emailing it to each other. The third is information routing. This is a newer area where we are now understanding that even glial cells can have conversations with multiple neuronal partners simultaneously, and glial cells can actually change the synaptic transmission level. So how do we map these networks? So multiomics approach basically lets us map intercellular communication pathways. So what you can do is you can pick a cell and you can look at all the different receptors as well as ligands that are there, and look at other cells and look at the corresponding receptors to those ligands that this cell is using. This will give us a kind of wiring diagrams between cells. Again, we are talking at a very micro level, and this would not be seen in any of the electrical signals that we usually see, which are large-scale electrical activities. So one of the insights about this via this multi-omics revolution has been about Alzheimer's disease, where we are looking at the molecular network failing. So one of the paradigm shifts in Alzheimer's disease has been, traditionally we used to think of Alzheimer's disease as basically a disorder of the neuronal cells, and when there is the disease which is happening, the glial cells will come and take care of the side effects of it. But the newer understanding is that the glial cells are not bystanders, but they are the central drivers of the disease process. This was revealed through single cell multiomics. So genome-wise association studies link top genetic risk factors for Alzheimer's like ApoE or TREM2 genes to predominantly genes expressed in glial cells, particularly microglia. This is genetic evidence which is pointing away from the purely neuron-centric model. So, how can the microglia affect the disease? So, first of all, these microglia adopt specific molecular states in the environment of amyloid plaques. And these plaques can actually induce a few genes which upregulate proteins for inflammation as well as phagocytosis. And finally, astrocyte and oligodendrocyte can also exhibit profoundly altered gene expression in plaques. So we are now looking at glia as basically the disease drive. Rather than viewing neurodegeneration as a neuron first problem with secondary glial reaction, we are looking at it as a simultaneous process. And that has led us to understand more about Alzheimer's, and we can now subtype according to the molecular activity we are seeing. So you could see something like an astrogliosis subtype where severe astrocyte proliferation and synapse gene dysregulation is happening, and this would be associated with faster progression and severe cognitive decline. Similarly, you are looking at subtype like lipid metabolism disruption, dysregulation of lipid metabolism within glial cells as a core pathology. In fact, this kind of echoes back to how Alzheimer had observed that there were adipose sacules inside the brain when he was looking at brains with AD. So even with our multi-omics approach, we are going back to the key things that were seen even before microscopy. Network vulnerabilities. So basically, now we are looking at a more active network where there is a lot of interconnectedness between all cells, and there is a two-way dialogue between these cells. Now, specific biological domains like immune response and lipid metabolism also acts as hubs in a molecular network. And any disruption in any one of these hubs can trigger cascading failures in the whole multicellular network. So a single dysfunction originating from a glial cell can actually cascade into severe dysfunction into the network. So that was the first layer or the cellular level. Coming to the second layer is the synaptic plasticity and neural circuit layer. So this is the omics to function bridge. Basically, when we are talking about memory, we talk about synaptic plasticity. And for synaptic plasticity to last for a long period of time, there needs to be long-term potentiation as well as long-term deployment. And especially in LTP, there are two phases of LTP. There is early LTP, which relies on transient changes, which will only last for a few minutes to few hours. This relies on post-translational modification of existing proteins, phosphorylation of AMPA receptors, rapid insertion into postsynaptic membrane, and increased sensitivity to neurotransmitters. But the main player is the late LTP or the long-lasting phase, which can last for hours to a lifetime. Now, this requires de novo gene expression, synthesis of new proteins, structural remodeling of synapses, and finally consolidation of long-term memory. So you can now connect from a molecular level to the protein level. For example, if electrical activity happens, there is going to be calcium influx via NMDA receptors. This calcium activates intracellular pathways, which actually opens up different paths to the nucleus. Now, these pathways can go and affect the gene transcription. CREB is one famous protein which can do it, and it activates immediate early Gs as well as late response genes in the nucleus. And finally, protein synthesis. So, new proteins are finally made from those switched on genes that we talked about, and these will actually go for the actual synaptic remodeling. Again, we talked about this previously, but it's a bi-directional communication. We are no longer thinking that, okay, axon comes, it gives it to dendrite, dendrite gives it to another one, and the chain goes on. We are looking at both of these having a active connection between each other and responding to the synaptic effect, along with the glial cell also playing a critical part. So the synapse is no longer seen as a passive recipient of molecular instruction, but a active participant that actually requests its own cell to give modifications from the genome based on computational need. So you're looking at this kind of a link: electrical activity which will trigger demand, which will be communicated to the molecular network via the pathways we talked about, which will produce a protein machinery to physically alter the synaptic network, and this will increase the synaptic strength and convert it into long-term memory. Another key change in our understanding has been about metabolics. So we used to think of energy as something which happens as a secondary thing. For example, in PET scans, we talk about how if a place is metabolically active, there is going to be higher amount of blood flow to that tissue, and that can be checked via PET scan. But we used to think that this was a secondary thing. The primary thing was the neuronal activity, and secondary to it, the blood would redirect ATP towards this specific area so that it can work its to its best possible layer. But when you look at the brain's energy demand, it is very, very high. You're looking at 2% of the body weight actually doing 20% of body energy consumption. And all of these are very high energy steps. So it's almost 100% ATP dependent. And the synaptic vessel cycle from neurotransmitter release to exocytosis to reuptake and recycling again consumes enormous energy. So now we are looking at metabolism as a dynamic regulator. Recent evidence basically suggests that it is not a permissive fuel but regulatory. In fact, specific type of metabolism is associated with different type of synaptic uh, effects, and there is substrate specificity as well as precise modulation of this metabolism according to calculational needs. So now metabolism can also play a part in the computational interface. So cellular metabolism translates the cell's internal energy status, a direct reflection of its integrated multiomic state, into neural computation. And one of the key implications that we came up with because of this is that any condition which affects cellular metabolism, from something like hypoxia to changes in the astrocyte neuron lactate shuttling to say mitochondrial disorders, can instantly alter information processing in neural circuits as well as long before any structural changes occur. So there's a sudden change depending on the metabolic state of the cell. One of the clinical insights we got via this concept was synaptopathy. So most direct clinical validation of this multiomics to synapse hierarchy comes from synaptopathy. And I'll give you an example. So this is GIRN2B gene. So in this case study, what we saw was GIRN2B encodes for glutamate N2B subunit of NMDA receptor, which is a primary molecular detector for inducing LTP. So what happens in the genomic layer is there is GIRN2B gene mutation. What this does to the proteomic layer is it will create dysfunctional NMDA receptors. What this will do to the synaptic layer is it is going to show impaired plasticity and circuit development. And finally, this will translate into a clinical phenotype of intellectual disability, developmental delay, and autism, epilepsy as well. So this framework has basically shifted the way that we can define diseases and classify diseases. And I would like to say it's a slow change, but sadly it's not. It's sudden jhatke padte hain unko. So that is what is happening. By understanding specific synaptic protein complexes and signaling pathways, and understanding which of them are disrupted, we can actually now make mechanism-based therapies to restore proper synaptic function. So this is how understanding about the synaptic layer has also changed our treatment of. The third layer is the bioelectrical grid. So the bioelectrical grid is basically coordinated activities of billions of synapses, and this gives rise to brain-wide bioelectrical grid characterized by rhythmic oscillatory patterns essential for cognitive function. And this is what we have to understand from sinuses to brain waves. Till this point, we were talking at a very small level. We were talking between cells. We were talking uh, between, say, 2 or 3 cells, not more than that. But for brain waves, you have to have 3 things happening. First thing is individual neurons have to fire action potential, generating small electrical fields. The second thing is a lot of these neurons, thousands to sometimes millions of neurons, have to fire in a coordinated rhythm. This rhythm is very, very important. And finally, these summated fields actually create what we call as the measurable oscillations, like what we see in EEG. So oscillations is also being looked at as a computational tool at this point. And what we understand is neural oscillations are fundamental to brain function, and they're not merely phenomenon. So they're not secondary to something. They're not uh, like a result of something, but in fact they provide mechanism for temporally structuring neural activity, and this is critical for various computational tasks. So this communication through coherence hypothesis suggests information is more effectively transmitted when sending and receiving neuronal populations and which oscillate in phase with each other. So, brief talking about the waves that we see in EEG, delta waves, characteristic of deep sleep, theta, linked to memory formation in hippocampus, alpha is prominent during relaxed wakefulness, and gamma is associated with active processing and attention. So, these are the rhythm generators that we'll be talking about. So, there have already been talks about connectomes. I will not go in depth into it, but I'm going to talk to you about a few key pacemaker topics. So just like in the heart, we have a couple of pacemakers, the SA node and the AV node. In the brain, we are looking at a lot of different pacemakers. Maybe around 20 to 30 have been already identified. One of them would be the suprachiasmatic nucleus. The suprachiasmatic nucleus, which is a nuclei in the hypothalamus, serves as the body's master circadian pacemaker. And this SCN is intriguing because it creates an intrinsic self-sustaining rhythm with approximately 24 hours period. And the function of the SCN will be it generates that intrinsic 24-hour rhythm. It will synchronize physiological processes to light and dark cycles, and it will control sleep wake cycle, body temperature, as well as hormone levels. This is based on molecular mechanisms like cell autonomous molecular clock, transcriptional translational feedback loop, and core clock genes like period gene or cryptochrome gene. So how does this work, this 24-hour cycle? So there is a negative feedback loop. When there is gene transcription, so per and tri genes will get transcribed into proteins. these proteins have a negative feedback on their own gene transcription. So every time new proteins are created, they will go back and switch off the gene, and it will have to be switched back on. This creates a kind of feedback which shows an oscillation and a 24-hour cycle can emerge. This provides a very good example of how molecular network directly generates the rhythmic electrical output that governs the physiological state. The other thing we saw about SCN was a lot of cellular diversity. There were a lot of distinct neuronal subpopulations using specific neuropeptides like prokineticin 2 to communicate and maintain robust network-wide synchron. Another one of these pacemakers would be the thalamocortical system. The thalamocortical system is the main reason we go to sleep and we wake up, and it comprises reciprocal connections between thalamus and cerebral cortex. There are three parts. First is the thalamocortical neurons. The second is cerebral cortex, but the layer between them is very important, which is the thalamic reticular nucleus. This is a sheet of inhibitory GABAergic neurons surrounding the thalamus, which is the master pacemaker for the body. So what happens in sleep-wake state transition? So intrinsically, we see that TRN and as well as the thalamocortical neurons create a rhythmic cycle. When the thalamocortical neurons fire, you will see excitation of the TRN. When the TRN fires burst, it will strongly inhibit the thalamocortical signals. This is going to lead to hyperpolarization of the thalamocortical ion channels. And once there is hyperpolarization, there is going to be rebound burst via the thalamocortical cycle. And this basically starts a cycle which keeps repeating. And this will generate oscillations and seep spindles, which basically disconnects the cortex from sensory world. So this is why what we are seeing in EEG while sleeping. What happens when you wake up? So neuromodulator input from the brainstem will now depolarize both thalamocortical pathway as well as the TRN simultaneously. This will break that rhythmic bursting that was happening due to a kind of equilibrium between these two. And network shifts from this tonic firing, shifts to a tonic firing mode to be basically ready for the whole day and faithfully transmits every sensory information to the cortex. But while you're sleeping, that network is cut off. How is there a molecular control of this beat? So this has to do with ion channels as well as transcription factors. Key pacemaker currents are, for example, P-type calcium current and pacemaker current. Now, P-type calcium currents are low threshold calcium currents responsible for generating rebound bursts crucial for the thalamocortical oscillation. And pacemaker currents are hyperpolarization activated cation currents, which provides slow depolarizing drive to initiate next activity cycle. And again, it's a dynamic interplay between these two, which creates those oscillations that we can see on ECG. Again, there is a transcriptional control of pacemaking. How is that? To give you an example, you can take transcription factor SHOX2. This is one of the master transcription factors regulated in the thalamocortical neurons. This goes and switches on ion channel gene expressions like HCN2, HCN4, and CAV3.1 channels. These channels, once this gene expression happens, they will you will see higher neuronal firing properties, and this will define the electrical characteristic of the pacemaker neurons. And finally, these pacemakers are going to control the circuit rhythmic function of the entire thalamocortical circuit. So this is a hierarchical chain of command that we're looking at. A transcription factor or molecular network controls expression of specific ion channel or the proteome, which defines the electrical property of pacemaker neurons and rhythmic function of the entire circuit. So this is connecting those three layer that we talked about till now. And loss of SHOX2 function can disrupt ion channel expression. This can alter thalamocortical firing, and this can lead to deficits in sensory processing as well as memory. One of the clinical insights in this layer is the uh, diseases of rhythm that we see in channelopathies and oscillopathies. To give you an example, let's talk about absence seizure. In childhood absence seizure, we see there is suddenly uh, brief episodes of staring, sometimes loss of consciousness. The patient will not remember anything which has happened during that period. And EV shows a very highly characteristic generalized 3 Hz spike and wave disturbance. Now let's talk about how this can happen from a molecular level to the circuitry. So one of the things we could find out was loss of function mutations of the SCN8A gene is specifically within the TRN neurons can alter intrinsic excitability and impair inhibitory connections. Again, looking at the different layers, in gene mutation layer, we are looking at SCN8A loss of function. In the molecular level, you are looking at dysfunctional ion channels. What is this going to do to the circuit? It's going to destabilize the pacemaker circuit. This is going to lead to pathological brain rhythms, which are the classic 3 Hz oscillations. And this is going to lead to a disconnect between the thalamocortical system, and it will lead to absence epilepsy episode. Other key ion channels we noticed are, so we talked about SCN8A, but there are also CAC-NA1X, HCN2, HCN4, GABR-V2, KCN2, Q and 3. These are associated with different types of epilepsy. And again, you will be able to trace from the gene to the kind of oscillation which is happening and correlate the kind of gene to function effect. Another key thing that we got from this layer is a better understanding about how to use something like deep brain stimulation. So this approach lets us think of DBS as an external pacemaker for the brain. And what we're doing is we are giving high frequency electrical pulses to the subthalamic nucleus. So it's working like a pacemaker. And this is disrupting the pathological rhythms or oscillations and restoring normal activity patterns. So again, plasticity and oscillation, we have a feedback loop. For example, learning will sculpt neural networks, and these neural networks will favor oscillatory dynamics because we discussed before, two cells when they are in oscillatory synchrony, they have a better chance of forming a better connection. And then rhythms will emerge, like structured oscillation arise from plastic changes. This will lead to more facilitated plasticity. So oscillation will provide optimal state for further learning. And this will again go to memory consolidation, which goes back to the same loop. Finally, the last layer or the brain-body communication network. And here we have to look away from thinking of the brain as a single organ which has minimal connections to the outer side. The brain is the master organ of the body. It has to have a intimate connection with every single tissue and hence every single cell of your body. And this is another key change in our understanding of the diseases, because we are now looking not only at brain disorders but also how these brain disorders will affect body functions and how body dysfunction or organ dysfunction in the body will affect brain dysfunction. So the brain commands line mainly via three different pathways. First is the central autonomic network, which is distributed network of cortical and subcortical regions, and it via the sympathetic and parasympathetic branches regulates all virtually every internal organ. Then you have the neuroimmune axis. So there's a constant bi-directional communication between brain and immune system. And brain regulates immune function. Immune signals communicate back to the brain. So this is one of the things that we got out of this was something like sickness behavior. So when you have a illness on the body, what that does is it will release cytokines. It will go and affect the brain's metabolism. That is going to lead to further dysfunction in the networks of the brain, and this we see usually as sickness behavior or malaise or tiredness, extreme lethargy when you're sick and sleeping more. Finally, there is a neuroendocrine axis, things like the HPA axis, HPT, HPO, axis. These also translate cognitive or emotional states into systemic physiological responses. So the central autonomic nervous system integrates internal as well as external information, and it ensures the body's physiological state appropriately matches the current demand and the challenges. HP axis, I'm not going to go into detail of. Yeah. So coming to clinical insight of this layer, one of the things that we got insights into is chronic stress as well as PTSD. Okay. So how does chronic stress create issues? So first of all, you will see persistent stressor, and this will activate chronically activate the HP axis. This HP axis is now going to give you elevated cortisol and prolonged high glucocorticoid levels. Secondly to these glucocorticoid levels, there is brain damage. And secondly to brain damage, there is impaired plasticity. When there is impaired plasticity, this again becomes a stressor for the brain. And this reinforces the pathology. So it becomes the vicious cycle. And cortisol's effect on the brain is not just structural. In structural, we see things like dendritic atrophy in hippocampus, spine loss in prefrontal cortex, reduced neurogenesis, and altered brain. But there is also functional impairment, like suppressing synaptic plasticity or LTP. There can be memory problems. There can be executive function deficits, as well as emotional dysregulation. So this maladaptive loop is thought to be a core component of the pathophysiology of major depressive disorder and anxiety disorder. Again, like I said, it's bidirectional. So we talked about how brain controls body, but we also understand how the body state can affect the brain state. So brain is not seen any longer as an insulated command center because it is continuously being shaped by the physiological state of the body it regulates. So hormones like cortisol and immune molecules like cytokines, they act on receptors within the brain, directly influencing gene expression, glial cell activity, neuronal stability, and synaptic plasticity. So the physiological state of the body, whether say inflamed, stressed, or metabolically compromised, is translated into a molecular and cellular state within the brain. PTSD is one example. You see a traumatic event where there is massive activation of threat circuitry and stress response. Secondly to that, you will see failed negative feedback to shut down the response. And this mechanism, once it fails, the brain will get stuck into a threat detection mode. So threat detection circuits are now overactive. This is going to increase the stress response, going to cause high cortisol, elevated inflammation, and this again is going to feed back into the cell, and this will actually reinforce amygdala activity, impair prefrontal cortex activity. Again, once it becomes a pathological loop where again it becomes a stress for the brain, and this loops continue. So the very mechanisms we talked about previously, which were good for short-term survival, becomes drivers for long-term illnesses. So these are few ways that systemic health can affect brain disease. For example, chronic inflammation, systemic inflammatory systems will alter brain molecular networks and increase neurodegeneration. Obesity, it works via metabolic dysregulation, affecting brain energy metabolism, which we talked about was also an active participant now, and this will affect synaptic function. Finally, metabolic syndrome, insulin resistance, and vascular dysfunction will also compromise brain health. So the health of the brain is inseparable from the health of the body, and we need to have understanding about this very clear. So just to again summarize what we talked about, there are four layers. The first layer is going to be the molecular multiomics layer or the cellular level, the what's happening inside the cell, inside the nucleus, inside the cytoplasm. The second layer is going to be the synaptic and circuit layer, where the neural connections and local circuit dynamics will take place. These two currently we cannot directly observe. The third layer is going to be bioelectrical grid layer. This layer is what we talk about as connectomes or different circuits. And this we will be able to appreciate via different functional scans, EEGs. And finally, the fourth layer is the brain-body interface, where there's a bidirectional activity of brain metabolism as well as body metabolism affecting each other. And like I said, it forms loops. And these vicious loops are basically a major issue that we usually see. Now, what this approach does is it gives us a much more clear idea about how cellular pathology can lead to large scale symptoms or large scale network dysfunctions as well as body dysfunctions. Secondly, we can also trace it back how large scale activity or large scale issues, say issues in your body with your metabolism, anemia, uh, some mitochondrial activity impairment, vitamin deficiency, is going to feed back into the primary cellular layer. It's going to change the cell gene expression as well as activity, and again create a kind of feedback loop. If the feedback loop is able to be broken by the homeostatic processes, the brain can go to the normal activity again. If not, it will raise to a pathological cycle as well as pathological symptoms. So the major thing that I really wanted to explain to you guys, or the take-home message was that first of all, that everything is interconnected. We are no longer thinking of areas in the brain, uh, looking at areas in the body. We are not even thinking of how the large-scale networks affect things. We are also looking at microcircuits and also intercellular circuits. So these are different types of circuits that we tend to not think of when we think about networks. And this is the reason I wanted to have this kind of a presentation for you guys. So that's it for the presentation. I'll stop my screen share and I'll invite any questions from the audience.
Thank you, Rutvik. A wonderful presentation. I think we need to read a lot. That is the take-home point number one. So,
Yeah.
over to chairpersons for their opening remarks.
Yeah, I agree to Dr. Aleem, sir. I think our own circuits and our own networks in our brain got a good kick out of this very stimulating discussion on the functional architecture of the brain. I guess there's a lot of things way ahead for us to explore and understand when we think about how a normal brain functions and how probably the brain of a patient with psychiatric disorder must be affected, understanding through the lens of this functional architecture. I have my own sets of questions, but before that, I'll ask Dr. Sri Gopal sir to please give his brief.
It is very lucid presentation, and as Alim sir has already told that we need to read lot of things to understand our brain. It basically sir has presented very in the nice manner regarding they described regarding the various layers of the molecular layers regarding the molecular multimonics, synaptic circuits, and bioelectrical grid, and brain and body interfaces, and also various diseases, which are how the molecular origin regarding the disease like the epilepsy, and the role of the deep brain stimulation in by explaining through the this molecular mechanism regarding the pacemaker of brain, like the sir as I explained the very descriptive manners. So I invite questions from audience regarding this.
I think before that, Dr. Parag, you can ask your set of questions as you get that honor first.
Thank you so much. So, sir, as I try to understand the topic, on one side, I am so happy to have more insights into understanding the human brain, but it also creates a lot of uh, exploring a lot of new things, making things more complex for me to understand. Like when I see all the layers or the networks that are interrelated, integrated from molecular to electrical to the structural networks, and every network is being influenced by maybe hundreds of factors for its functioning, and it's a dynamic thing which keeps changing every time. So I would like to ask a question that how far we are from having diagnostic criteria getting confirmed based on some diagnostic tools, maybe some investigations, maybe some brain scans, maybe some EEG records that are able to tap exactly the locus of the pathology structurally, electrophysiologically, or molecularly, and help us to define that illness or differentiate it from the other illness. So when are we looking forward to translate this basic science into clinical practice? How far we are? That's my first question. And second is that the same way, how far we are expecting the intervention model or treatments, which are focused precision-based medicine, targeting this functions of the brain and helping us to help our patients. Yes, sir.
Okay, sir. If it's okay, I will just share my screen. I want to show you guys one lecture I did just previously to this. It was for a government hospital in Noida, and they wanted to understand how genetic testing is letting us understand neurodevelopmental disorders and whether there are any kind of um, yeah, like therapeutic approaches we can do. Okay, so I'll just share my screen. Yeah. Okay, so just a quick glance. So I'm not going into detail about this. What we are understanding is we have previously, when we are looking at developmental disorders, now I'm mainly speaking about developmental disorders because me being in the child area, we see these quite a bit. And previously, what we used to look at was karyotype. Then we came to what we call as the chromosomal microarray, which gives us a 15% yield. But currently, we have come into what we call as the next generation sequencing revolution and the multi-omics approach. Now, this multi-omics approach is actually letting us have a 35% minimum diagnostic yield. And what I mean is, what we need to do to be able to come to that level where we can identify disorder via genetic markers, which is what is happening in onco, is that we have to have these kind of sequencing. And only with genetic sequencing are we able to come to that level. This also not only is giving us a chance, this not only is giving us a chance to understand how developmental disorders are, but a very interesting thing that we can now do is use computational models to predict what kind of issue will happen due to a specific disorder. And there is now technology called the induced pluripotent stem cell technology, where what you can do is you can collect a cell from the say mouth or say blood. You can reprogram that cell into an embryonic like pluripotent state. You can then differentiate it into specific neurons, and this is all from the patient's cell. And with this, these you can make different models, and they're already been successful in a lot of places. For example, Rett syndrome, MECP2 model has actually worked. In Fragile X syndrome, FMR1 gene modeling has actually worked, and these not only catch the cellular phenotypes but also predict the kind of disease or disorder which is going to happen. So another interesting part about this is we can now create these omics and model systems, and there are now treatments. Okay, so this is, for example, targeted therapeutics that we are looking at. For example, in tuberous sclerosis complex, we see TSC1 or TSC2 mutations. Treatment is now going towards mTOR inhibitors, which are very specific towards this, which will reduce tumor size and treat refractory epilepsy. Rett syndrome, you're looking at something like trofinetide, which reduces neuroinflammation and improves synaptic functions. Fragile X syndrome, you're looking at something like mGlu5 antagonist or say metformin or low dose sertraline, which can lead into early language development. I'll stop my screen share. So yeah, we are not very far. We are already on the pathway, and whether we like it or not, we are going to get there. What we are trying to do is make sure that we are on that same track, we stay on the same line. Now, this kind of an approach is being used in neurodevelopmental disorders, but it can be extended into adult disorders, it can be extended into geriatric disorders as well. And this is why it is so interesting that, see, science will go forward whether we like it or not. And because of the internet revolution and after the pandemic, the level of connections that everyone now has via WhatsApp group and whatnot, the flow of information is way higher than anything that our predecessors have seen. We no longer need to go and look at different research articles. I can easily now pick up a AI and ask it to summarize it for me. And I can look at AI in a way that it can do a computational thing based on these genes. There are various models which have been successful in various diseases, including neurodevelopmental disorders, and we are probably not very far from targeted therapeutic approach. So again, the advantage in onco was once they understood this kind of a molecular approach, they were able to make specific specific antibodies or kind of designer drugs which were targeting only these specific gene modulation. And we are now slowly coming to us, but we have to understand that in onco you are looking at one gene mutation, two gene mutation, that's it. You're not very worried about cell to cell transmission and everything else. While we are looking at polygenetic multisystemic diseases, and polygenetic multisystemic diseases are probably going to be the toughest thing to approach via this model. But the advantage is we finally have the computational power to be able to do it. We can model these things now. We can connect it to actual phenotype, and once we are able to match it properly, we have a proper disease that we have identified. It is no longer a disorder; it is a disease that this specific gene mutation is causing this kind of a phenotype. We will find different phenotypes within that gene function pool, and finally, targeted therapeutics will also be available because we now understand these kind of molecular mechanisms.
Thank you.
Hope that answered your question, sir.
Yes.
Yes, sir.
Ritvik, I think a very comprehensive presentation. How much lack of biomarker to our like phenotypes, which basically the approach of a clinician is phenotype based treatment, symptomatic treatment. So lack of biomarkers add to the confusion, which you are saying. And as a clinician and practicing in a chamber, so what I can do about it, how this presentation helps me.
So, one thing that it will immediately help you in is any kind of developmental disorder. So, when you're looking at specific disorders, for example, where you have very clear genetic history, you're seeing uh, like genetic traits in the father's side or the mother's side, you can go with further genetic testing in the child. And once you get the gene testing done, you can do a modeling of the phenotype, and it will actually predict. So one of the things that we were able to do is this kind of a thing. I think Dr. Bhaskar already does it without the help of AI, but for us normal people, we'll probably need a computational power like AI to be able to do it. But it is easily something that can be done, and at this point, we are already there. I'll give you an example. We identified a case which was a variant of Smith-Magenis syndrome. I'm forgetting exactly the uh, gene that had come up. Now, this case had gone via various different gene testers, and the like gene testing, once he had gotten it done, it came up as variant of unknown significance, which is typically the picture you will see if you ever send any of your patient for gene test. And we were able to match that variant of unknown origin to a variant of Smith-Magenis syndrome. Now, this kid had come to us with phases of like um, issues in executive functioning as well as speech development. So, kind of a picture of what we call as autism. Now, via this model, we were able to predict that this patient also has a very high risk of having epilepsy because the gene that we were looking at where ion was basically a channelopathy, so ion channel issues were there. So what happened was this patient was kept on I think one of the antidepressants as well as methylphenidate. Now, for all of us who are well into child disorders, when I was first trained in giving methylphenidate, we were always told to look for history of epilepsy. Do not give in a child with epilepsy because it can sometimes precipitate seizure, is what we used to think. But with this kind of a understanding, we already knew that this kid is going to get epilepsy, whether possibly, and whether we give methylphenidate or not, it is possibly going to happen. And this did happen. The child did have a like epileptic seizure later, and he was put on anti-epileptics. But we were ready for it. The parents were already educated about this, and we were ready for it. So we did not get scared about methylphenidate causing any issues. We just added a anti-epileptic, and methylphenidate was continued. Patient responded really well to this line of treatment. This would not have been possible without doing this kind of a matching. Again, although right now the testing from a genetic perspective is still costly, but the previous presentation I just showed you, this was done for a government hospital in Noida, who are now starting gene sequencing. So they are starting 3 gene sequencing in Noida, GIMS I think, and they are making it available for clinicians. So soon we should be able to get these kind of resources because see, at this point it is more about gene mapping. We are trying to understand disease from a gene to phenotype perspective, and if we can match that kind of a thing, this will let us identify direct diseases. And anytime next time a clinician faces that kind of a picture, he'll know exactly what he's facing. He will also know exactly what kind of molecular treatments are available. He will also know what kind of treatment was gotten from there. And this again, to just give you an example, let's say we are picking up autism. So what we call as autism, we are looking at maybe a few millions to possibly a few, maybe even a billion different combination of genes which are interacting with each other and causing these symptoms. So if we can identify each of these and we can test and put the patient into this category that will let us understand, predict not just his current symptoms, but also what is going to happen later. For example, in this child that I was talking about, we are now able to map what will happen probably in adolescence. We are now able to have an idea about the trajectory in adulthood, middle ages, and possibly a trajectory in uh, old age as well. So we are shifting from a kind of symptom uh, classification or disorder classification to this kind of a trajectory approach that we usually validate via genetic testing. And if we can actually identify these trajectories, this is probably the best thing we can do in psychiatry. Because once you have identified the trajectory, it's mostly going to be pretty much the same. You are not going to uh, face the kind of issues that we currently face. For example, uh, in child, we are facing way more genetic mutations compared to previous generations. So when we are looking at patients in the geriatric age groups, the adulthoods, as well as children who are just born now, the level of mutations are different. The mutations in children are way higher because way more genetic variations are seen in these children. And again, if we uh, like, I face this kind of a challenge where you think that you're supposed to calm the child down, you give him clonazepam, and he goes completely berserk. I think a lot of people among us would have seen similar cases where you don't expect that this kind of a response can happen to clonazepam. Clonazepam, if I give it to any of the elderly people, I know most of them will actually calm down. Very few people will get agitated. But when in child, it's a huge number. Like I'm looking at maybe say 15 or 20% where they will give this kind of an atypical response to the medicine. And it's very hard to figure out what it is. With this kind of an approach, we'll be able to identify these trajectories. We'll be able to even say that this, if you're going to give this kind of a molecule to this trajectory, is going to have this kind of a response. So again, a lot of clinical implications is out there. We are just at the beginning of this era, and this will keep going on, is what I understand.
So you're saying overall the gist is that you will go for a genetic test, right, Prithvik?
Genetic test is going to be one layer.
One
So
layer.
see,
The starting
genetic,
point, like starting
yes,
point, and then when
probably
to
the
do it.
most important.
1, 2, 3, 4 scenarios when to do it.
So again, when you're looking at specific cases where you're very clear about the genetics part of it, like I'm also talking about cases like geriatrics. So I observed with a genetic uh, tester. So what they like, even in geriatrics, we see something like if there is a very strong history of say something like frontotemporal dementia on one side or the other, gene testing actually picks it out when you're doing it even for a geriatric person, and you will be able to predict what kind of disorder he will be going for. The other thing is this is just one level of or one layer of information. There are other layers of information, so we need to integrate all of these layers. So this first layer is probably the most important because again, everything molecular is going to have a large scale application on the large side as we go to the macro side. But similarly, every single layer has a different approach. So the first layer is more about genetic testing. Second layer again, more about genetic testing. Third layer, you are looking at things like EEG, functional scans. You're also looking at the overall symptoms that we are looking at, which are basically outputs of large-scale neuronal networks. And you should be able to integrate all of these. So you're not just looking at, say, a symptom like a patient when he comes to you. Ideally, you should be looking at not just his current symptoms, but also you should do what we call as phenotyping. You should be able to identify what kind of phenotype he has. Phenotyping is already there, where we don't need to do gene testing. For example, there is this app called Phase2 Gene, where you just upload a picture, which will give you an idea about what kind of genetic disorders there are. Because again, different structures of the like brain are developed in different ways, and these somehow correlate with different structures of the body. And if we can find out these, we can do a kind of overall phenotyping which doesn't require gene testing. But yeah, integrating that also would be very helpful. So again, you're looking at different layers, and if you can think of diseases from this different layers perspective, you'll be able to not only identify the kind of picture that you're looking at, but also you'll be able to identify what kind of therapeutics would be possibly better for you. And it also opens up a lot of things. Like just I showed you metformin usage in patients with few patients with autism. It could actually increase speech development. We could not have guessed it unless we knew the mechanism of what is happening.
And I think galantamine also, I think one of the molecules which they have brought out. But I think Vihang sir wants to ask about that you have compared the scenarios with oncology. It is important to note that every case of multiple myeloma marked for gene therapy is treated with individualized gene therapy, which cost several lakhs per injection. Is this applicable in mental health care also?
See, I will give you an example of say anti-epileptics. So when you're looking at something like sodium valproate, which was one of the first anti-epileptics out there, it was something which was working on everything. But again, take a look at the anti-cancer drugs. The like in my dad's generation, when they were treating onco, they were basically being given cytotoxic drugs because the point was to delete whatever rapidly uh, evolving cells are there, and it would take care of the cancer, but obviously it would also cause issues in skin, hair loss. It would cause GI issues. And now we are now at targeted therapies where we are actually looking at people who go through chemo without having major problems in stomach, skin, or anywhere else, mainly because there are targeted therapies. Are these targeted therapies costly? Yes, because currently the approach that we are taking is a high cost effect approach. So we are making um, we are making specific antibodies and we are attaching it to specific molecules and we are using those antibodies to go and do the work. This will ensure that it will only target the specific like tissue that you want. But the cost of making this and creating this is going to be very high. So one of the things that AI is revolutionizing in like medicine is actually repurposing of things. So one of the things, for example, I give you that example of metformin. So one of the things that we are now looking at is repurposing of the older drugs. But now we should first look at what molecular level effect that drug does, match it with the kind of molecular effect you're expecting in this patient, and give it according to that. With that, you should be able to have a low cost, high effect approach where you are not really testing. So again, you're looking at different layers. You're trying to integrate phenotypes. You're trying to integrate trajectories. You're trying to integrate things like the symptoms or large scale networks. You're trying to integrate PET scans. Along with this, uh, let's say whatever we have possible, maybe we just have EEG at our hospital, government hospital, EEG है हमारे पास. Fine, EEG है हमारे पास, we can do phenotyping along with it. We can also map the symptoms. We can at least integrate these three layers. These three layers की वजह से we should be able to identify specifically the synapses पे क्या हो सकता है. We can make a hypothesis in our brain about this patient कि this patient में ये वाला साइनेप्टिक इशू हो रहा है इसके वजह से इसके लार्ज स्केल नेटवर्क में ये वाला इशू हो. अब इस वाले साइनेप्टिक इशू होने के वजह के कारण मतलब probably this kind of cellular mutation or gene mutation is going to cause this. If we can trace it back like this, we can now attach a specific drug to it. Even right now, most of the like drugs that we are having, they are at the level of वो previous वाले cytotoxic levels के जो onco का treatment है. So here, what we are looking at is repurposing all of our drugs in this manner in a specific molecular pathway. And once you are able to repurpose these drugs, again, the cost is going to be minimal. You can look at something like using escitalopram or amitriptyline for autism, which we are already doing right now. and we have had very good results with these kind of approaches. Very low cost. It's only in places where there is not a very quick answer that you get via these
Yeah,
layers.
Ritvik,
That
I may help you. A very short and direct answer to YS's questions. For
Hmm.
targeted therapies, you need targets.
Yes,
Mental health is actually not the target. What we are mental health, what we are seeing is a cluster of syndromes, symptoms occurring together. So it is like bringing Greg Chappell and teaching everyone to be right-handed batsman and a left-handed bowler. So what targets we are choosing, that we have to revise. So again, we have to
yes,
move away from ICD-DSM, look at what are the hundreds and thousands and millions of the uh, molecules and the genes that are causing the symptoms. So basically, what we are seeing mental health right now is just a superficial overview of symptoms. So when we move away from that approach, this presentation comes in. So you target because in cancer you have one gene or two genes and then you target that. So targeted therapy is possible where there is a target to identify. You cannot shoot a Brahmastra to do anything. So I think that should answer YS's questions.
yeah. Sir. Actually, that is perfectly put. Much better than me, I would say.
And I think Ritik, your question, your presentation challenges this software, hardware. I think a very erroneous distribution or the distinction on neurology psychiatry. So there is a question in the chat box. So is merger due or imminent?
See, what I would say is the more I go into this, the more I realize that the ideal doctor is not a specialist or a super specialist. Maybe a ideal surgeon could be one, but ideal medicine वाला जो है वो general practitioner होता है. He should be able to identify not just कि brain में क्या चल रहा है, but बाकी जगह क्या चल रहा है. So leave alone neurology and psychiatry coming into one. Maybe we might see soon uh, a complete body approach. So you're looking at integrating all different system knowledge into these. I did not go into part of this presentation, which was the drugs that we're using. We जो हम देते हैं, let's say you're giving something like escitalopram, and we think कि उससे brain में क्या होगा, उसकी वजह से क्या होगा, fair enough. It's a good way to go about it. But diffusion is going to happen. It's going to go to every other cell of your body. And this approach will let you understand what kind of effect it can have over different other organ systems as well. One of the key insights with this was, uh, this is more of a personal, I would say, insight from my side. I gave escitalopram to one of the patients who had company for depression. She was 80 plus and she had a lot of blindness. She was only able to distinguish between dark and light. So the approach there was escitalopram for depression. But क्या हुआ? The patient got her eyesight back. She was able to start looking at it. So I started thinking maybe next time I see something with retinal pathology, try करके देखते हैं. So next time I did see something with retinal pathology, I started doing it. Then there was and I told the patient कि ऐसे होने वाला है, look for the improvement. उसमें improvement दिखने लगे, not in everyone, but in a majority, I would say. Next thing that I started thinking was maybe if this is working for retinal disorder, maybe we could look at something like sensory neural hearing loss. उसमें देना शुरू किया, tinnitus में देना शुरू किया, उसमें improvement आने लगता है. Next thing was like I was like यार अगर brain और उसके related में कर रहा है, what about heart? What about liver? What about lungs? So one of the patients में liver dysfunction था, उसमें दिया. Liver functions started getting better. So again, our
Again, Ritvik, a word
limitations.
of caution
Yes.
here. Ritvik, a word of caution here because again we are on a public platform. So basically, there are courts, there are consumer forums. So again, before trying all this, we have to think
No,
twice
no,
about
sir,
all these
I am
things.
not talking
Yeah.
specifically about giving it in these disorders. I am talking about giving it in patients जहाँ पे हमारी symptoms है ही और उसमें ये disorders हैं. Look for the improvement in other places as well. If you don't look, you won't find. That is what I mean. It's not like I would not never
Absolutely.
recommend
And
giving this kind of thing.
yes. So it should be teamwork. Physicians
Yeah.
or other faculties should also be involved, and we should not cross any ethical boundaries in doing this.
Yes.
Regarding Shobhit's question, I think merger is still okay. Takeover and coup d'etat are not. So I think what Ritvik is trying to save us is from the coup that psychiatry may be taken over by some other branches. So thank you, Ritvik, for bringing us back to basics, most of the basics. So Ritvik, the thing is, you talked about 4 layers. So initially there are molecules, then they are combining to form the synapses and neuron. Yeah. Then the next layer would be.
The large scale network that we're looking at, or the different circuits that we're looking at. And then finally would be the brain body thing.
Okay.
And one interesting
Okay. So
paradigm.
I think this is the
Yeah.
this will be the crux of the matter that we have to. I hope you're not getting the ringtone. Yeah. So the crux of the matter is this. So from molecules to we are going to behavior. So now this stretch of imagination that we have to cover. So our ICT is just on the superficial level. Now in this deep level, or what Ritvik is trying repeatedly to, and our other members of Life Shemi, we can say, we are trying to do just that. So yeah, Vishal, over to you.
No, I was just saying that one paradigm I understood, which was very new for me, was you're saying even body is controlling the mind, which is a good paradigm. Usually we talk about a top-down approach. This is not top-down; it's two-way approach.
Yeah, absolutely, it's two-way.
So that's
So
an
I think in
interesting.
general, bidirectional everywhere is what I understand.
Yeah.
So overall, sir, this establishes the lack of biomarkers are creating the whole confusion, and probably this presentation is for defining the biomarkers, which might take maybe how much of time we don't know.
But multi-omics approach is allowing us to get to there.
But we don't have, we have, Ritvik, any biomarker as of now, which is in the stages of some kind of because BSNPs did it, tried to regulate it, but again, no definition of a biomarker is there in psychiatry till date.
Definitely, there is definitely a lot of biomarkers जो अभी आ गए हैं. And for example, जैसे I showed in Rett syndrome, you directly check for MECP2 mutation.
Again, it's a monogenetic. It has a defined gene. That is what again.
Got it.
But before we go to biomarkers, we need to first redefine our disorders itself.
Yes.
Basics, we need to look at that also. There's also an approach going on, right? We still don't know what is schizophrenia exactly. What are we looking at there? It's been tried redefined. Mood disorder has been redefined. So I think
biomarkers, we are mainly now shifting from looking at large scale biomarkers to cellular biomarkers. And cellular biomarkers is probably going to be the key in understanding and treating these disorders.
Right. So I think we should start winding up now. It's 9:30.
Yes, please. So
with your permission,
that we have left? Yeah.
Yeah, with your permission, may I summarize?
Yes, sir.
Yeah.
आप ही पे आ रहे थे हम. आप ही कर दो.
Okay, thank you so much. So I think the gist of today's discussion, which was very, very thought-provoking and intellectually stimulating, is that while our modern neuroscience and the mental health science is progressing towards finding out more and more brain mapping based raw data that we generate in our hospitals and give it and fed it to AI based computer models, computational models to work on it and help us to identify specific biomarkers and probably link it with specific clinical conditions. While this process goes on, while we keep collecting the raw materials from our patients through brain mapping, keep feeding it to the computers, and maybe one fine day we would have a strong association with some disorder with some biomarker so as to set a stage for a molecular biology laboratory to start working on designing intervention biological models or electrical models for treatments based on those focused precision identified pathologists. Till we achieve that, I think we continue to work based on our conventional wisdom of last many decades, which largely rely on randomized controlled trials based on identifying the phenotypical or the symptomatic presentations of thousands and millions of patients studied over 20, 30 years, and the overall wisdom and the clinical impressions and experiences tested against the randomized controlled trial benchmark that proves and that helps us and guides us today in our clinic. Thank you.
Perfectly summarized. Thank
Very
you.
well, very nice. Very well
Dr.
summarized.
Sri, any comments from your side?
You're on mute, Dr. Sri.
Yeah. Sir already has summarized in a very lucid manner, and Dr. Ritvik has presented very well and answered all the questions, sir. There's no comment from his side.
Thank you.
Thank you, sir. Tufan sir, over to you to end the session.
Thank you. Thank you very much, Dr. Ritik, for an amazing picture of what is going to come and what may be our treating strategies or successors treating strategy in times to come. And we realize the limitations, but we appreciate the changing picture because change is the only constant. And I sincerely thank you. I thank Dr. Shobhit and Dr. Vishal and Dr. Amrit for helping this program, as always, as the vibrant moderators. Dr. Parag, who is regular to this forum, has made a good beginning and end, and Dr. Sri Gopal's contribution is very nice. One thing I suggest, as was told by Ritik, this becomes the right and of every doctor who is treating a human body to know and be oriented in this way, not necessarily neuropsychiatrist. To make it possible, we must incorporate this in the undergraduate education orientation towards this. What I find it is not there. How you become a specialist, you become oriented in genetics, you suddenly find that these are the things, but it will take time to evolve. We can expedite if we can orient the students to this. That's all. Thanks, everybody. Thanks, Darren. Thanks, all the participants. Good evening and good night.
Thank you, sir. Good night,
Thank you
everyone.
so much.
Summarized with a rider. That was the best thing.
Very nice. Thank you, Dr. Parag, Dr. Sri.
Thank you.
Yes,
Thank
sir.
you. Thank you, Dr.
sir.
Ritvik, as usual. Very nice
Thank you so
presentation.
much, Dr.
Thank you.
Ritvik.
Yes,
It was a
sir.
pleasure listening to
Thank
you.
you.
Thank you.
I'm closing the meeting.